Hello and welcome back to Excel Share Academy. So now we are going to do the second part of the IIT JAM 2019 Biotechnology explanation for the questions. Okay, so this is the second part in which we are going to do the another 10 sets of questions. So your 11th question is, it's belonging to the maths, limits and derivatives. So if so the question is, let A is equal to root of 5 plus 1 by 2 and B is root of 5 minus 1 by 2, then limit n tending to infinity a raised to n plus b raised to n divided by a raised to n minus b raised to n will be what? So for this, this is the way we are going to solve it now. So for this, you need to know what is the answer for this, right? So what we are going to do is that, let's see what is a by b first. Okay, so what we are going to get, root of 5 plus 1 by 2 again divided by root of 5 minus 1 by 2. So 2 cancel. What we get is root 5 plus 1 by root 5 minus 1, right? Now, the whatever value you put, if you know the value of root 5, what we see that this whole entire entity is going to be greater than 1, right? Now, similarly, if I do b by a, so what I'm going to get? Just the opposite, right? So root of 5 minus 1 by root of 5 plus 1 I'm going to get. So now this entity is going to be less than 1. Okay. So if this is clear, yeah. So now we know that a by b is greater than 1 and b by a is less than 1. Okay. Now what we are going to do is, let's see what limit we are getting now. So limit n tending to infinity a raised to n plus b raised to n by a raised to n minus b raised to n, right? Now this can be written as, like, we are going to take this form. So this will be a raised to n, take the common, 1 plus b by a raised to n divided by a raised to n, 1 minus b by a n, like this, right? So now what you can do is cancel. So now what answer you're going to get? So we know that the value is of b by a is going to be less than 1. So which is negligible, right? So this can be written as 1 plus 0 divided by 1 minus 0, since this quantity is going to be negligible. So now what I got is that 1 by 1 is equal to 1. So here your answer is 1. Answer option A. Okay. So this is how you're going to clear. This is how you're going to get the answer for such questions. Now moving on to the next question. So this is again mathematical question only. So in how many ways can one write the elements 1, 2, 3, 4 in a sequence x1, x2, x3, x4 with xi not equal to I for whole numbers of I? Okay. So this is also very simple actually. Now the elements here are given as 1, 2, 3, 4, right? So this can be written in 9 forms. Now how it is? If you see 1 by 1, 1, 1, then you have got 1, 2, similarly 1, 3, and then you have got 1, 4, right? Then 2, 1, 3, 1, and then 4, 1. Now we have 1, 2, and 3. Last 1, 2, 3, and 4. So 4, 3, 1, 1. So how many total? Total we are getting 9. So the answer is option A again. So I hope this is clear. Let's move on to the next question. Again, another mathematical question. This is belonging to your trigonometry. So the question is given, sin A by 1 plus cos A plus 1 plus cos A by sin A. So you just need to simplify it into one singular entity. So for this, let's begin. Okay. So yeah, so this can be done. This is also very simple. Many methods are there. So one of the methods, I'm going to share it over here now. So what you can do is take the LCM now. Okay, so what will be the LCM over here? This becomes sin A into sin A plus 1 plus cos A whole square. And this becomes your denominator, right? Now, what we also know that what is 1 plus cos A square? 1 plus cos A square is going to give you 1 plus cos square A plus 2 cos A, right? So this is what you're going to get now. Now put this value over there. So what we get is sine square A plus, put this value, 1 plus cos square A plus 2 cos A. Okay, divided by 1 plus cos A sine A. Now we also know that sine square x plus cos square x is equal to 1. So we can take this value and this value. So this will become 1 plus 1 plus 2 cos A divided by 1 plus cos A sine A. This can be written as 2 plus 2 cos A by 1 plus cos A sine A. Okay. Now just let me have some space. Okay. So now this can be written as take 2 common. 1 plus cos A by 1 plus cos A sine A. Simple, cancel, cancel. What you get is 2 by sine A. And 2 by sine A can be written as 2 cosec A. So your answer is B. So I think this was useful and easy also to solve, right? So let's move on to the next question. Okay, so this question is from Bio. The evolution of eyes in octopus and in human is an example of dash. So what will be the answer? So before we know the answer, we should know what these options are meaning to say. So the first option is divergent evolution. In case of divergent evolution, it occurs whenever two species are evolving differently, like one from here, but from a common ancestor. For example, we know about the Darwin finches. Okay. Now coming to the convergent evolution, so it is a process where organisms are not closely related, but independently they evolve similar traits which they evolve because of the adapting to the similar environments. So that is, even though there are species A and B, they are growing differently. Since the habitat was similar, so they develop the similar organs. So for example, the camera eyes of the octopus is very much similar to that of the humans. So phylogenetic and embryological studies suggest that the camera eyes have been acquired independently. So the eyes of octopus and humans have independently evolved after the divergence of the two lineages. So that's why they are considered under the convergent evolution. So from this, we know that for this question, the answer is convergent evolution. But still, let us learn what are the rest of the options say. So adaptive radiation, it is relatively a fast evolution of many species from the single ancestor. So for this, the option is Darwin finches, if you have seen in the Galapagos Island. So from one island, when it got split into two, it's not two, many other islands. So there, whatever the kinds of birds were present, they develop according to the fruit or nuts available or the flower available, they develop their finches. Now, what is genetic drift? It is the mechanism of evolution in which allele frequency of a population change over generation due to chance events. So now we know all the meanings of these four options. So from this, it is very much clear that it is talking about the convergent evolution. Okay. So again, moving on to the next question. Which one of the following modification occurs both on DNA and protein? So this is quite simple actually, because see, first of all these modifications are happening on the protein. Okay. So only one thing what you need to remember is that the modification which is also happening in the DNA. So among all these, if we see the options, ADP ribosylation means the addition of ADP ribose group to the protein. So this is again a post-translation modification which is happening in the case of the protein only. Now methylation means addition of methyl group. So we also have heard that during DNA also, in the DNA also there is a addition of methyl group, and that becomes your CpG island also we talked about. So if you learn more about DNA replication transcription, there you will talk about the methylation, DNA methylation, that is which will help in differentiating between the parent strand and the daughter strand. Okay. Then simulation and ubiquitination. So ubiquitination, you know that the ubiquitin molecules are added to the protein. These molecules are added to the protein so that they are targeted for the proteolysis. Okay. In the same way, simulation is also the analogous of ubiquitination, because in this case also small ubiquitin like molecules are, that is called SUMO. That is, small ubiquitin like molecules are added to the protein so that they are again targeted for the proteolysis. Okay. So from all this discussion, it is very much clear that the modification which occurs on both DNA and protein is the methylation. Okay. So let's listen about this. So protein methylation and DNA methylation, it is an important post-translation modification using cellular signal transduction pathways. Now, proteins can be methylated at both the ends of N and C terminal and on the side chains of the nitrogen of the arginine and lysine residues. Please remember, so on protein, it is the lysine and arginine amino acids to which the methyl group is being added. Now, the ORIC contains the GATC sequences. It contains 11 GATC sequences, that is the CpG island which I was talking about. So that gets methylated on the adenine on the both strands. So only a fully methylated origins can initiate the replication, and hemi-methylated cannot initiate the replication. So from that, at the end, you know that only the methylated DNA strand, that is the parent strand, and the daughter strand will not have any methyl groups. Okay. So remember the sequence. So the sequence is GATC, to which adenine group, methyl group is being added during the replication of DNA. If any doubts are there, please comment me, or you can message me in the Telegram by the name Excelsior Academy, and you can ask any questions if you have. Okay, moving on to the next. So solutions of the following peptides are prepared separately at a concentration of 1 millimolar. Among these four, which one has the highest 280? So in this, it's very simple actually. You just need to remember that whenever you talk about A20, that is absorbance at 280 nanometer, and if it is talking about the peptides or proteins, you just need to remember that it is always the phenyl, tyrosine and tryptophan which actually contributes to the absorbance. So if you have got more number of these aromatic amino acids, these are called aromatic amino acids. If you have got more number of this, that particular solution is going to have the maximum absorbance. So let's see which option is having. So if you see in this case, you have got one trypto, one phenyl and tyrosine and tryptophan. Okay, here 4 are there. In the second, 1 and 2, only 2 are there. Third option, you don't have. Okay, 1 is there. Last option, only 1 is there again. So among all the options given, we see that option A, this particular peptide has got high number of aromatic amino acids. Hence, the answer is A, which will have the highest absorbance at the 280. Now, the free energy required to synthesize a mixed anhydride bond of 1,3-bisphosphoglycerate is generated by the oxidation of dash. Okay, so if you see that in this case, aldehyde to acid, alcohol to acid, alcohol to aldehyde, or NADH to NAD+. So in this case, if you see the reaction, should I make the structure? Okay, let's make the structure over here. So this is your glyceraldehyde 3-phosphate. So that's how it looks. 1 second. This is your phosphate at the third carbon. So this is your glyceraldehyde 3-phosphate. Okay. Now, whenever there is a glyceraldehyde phosphate dehydrogenase over here, then what you get is 1,3-bisphosphoglycerate. So this is why I used to tell all my students that you need to practice the structures also, so that it is easy for you to answer during your exam. This is your 1,3-bis phosphoglycerate. So what do we see over here? Over here, the NAD plus is becoming your NADH, whereas option is given NADH to NADA plus. So that's why it is not, right? And option one more phosphate group has been added. You can see over here. Oh, sorry, I didn't make it. Okay. So one more phosphate, that is 1,3-bis phosphate, right? The 2 phosphor group is going to be present over there. So with this reaction couples an oxidation step within a phosphorylation step. So what do we see over here? That acetaldehyde is going to become a acetic acid. So option A is the correct answer over here. Okay, so moving on to the next. The following reaction is an example of dash. So if you see it carefully, you will see that it is what? It is the example of isomerization. Okay, if you see the structure carefully, it is the example of isomerization. So isomerization is a process by which one molecule is transformed into another molecule which has exactly the same number of atoms, but the atoms have different arrangements. So if you see same number of carbon, same number of hydrogen, same number of oxygen will be present. So the given reaction is an example of isomerization. Now, which one of the following parameters changes upon doubling the enzyme concentration? So, according to the Michaelis-Menten kinetics, if the concentration of the substrate is increased more, the enzyme molecules will work efficiently. So, if enzyme concentration is double, the Vmax will be double. So, where Km, it is the value of the substrate concentration at half maximum velocity. It is independent of enzyme concentration and remains exactly the same. So from all this information, we understand that option B is the correct answer, that whenever you increase the concentration of the enzyme, that is you're doubling the enzyme, the Vmax will also be double. So answer is Vmax over here. So for this, you need to study the Michaelis-Menten equation of the enzyme kinetics. Okay, so which one of the following statement is a correct description of modes of action of taxol and colchicine? So actually this taxol, colchicine, they actually stabilize the microtubules and then they do not allow them to form the spindle fibers, and hence it is arresting the cells at the M phase. So if you see all the options over here, the answer is going to be the taxol, which actually stabilizes the microtubules and it prevents the microtubule formation, and because of which what will happen, you know that chromosomes are there and microtubules are not able to bind to the chromosome. So what will happen? It gets arrested at the M phase only, that is the mitosis, in the metaphase. And we use certain these molecules in order to study the cell cycle or the chromosomes of the cells. So that's all for today. I hope you like the video and all the questions were actually explained beautifully. So if still any doubts are there, you can message me and ask me in the Telegram also, as well as in the YouTube comment section also. So thank you for watching. And the another session, that is the third set of this question series, will be uploaded soon. So keep watching, keep studying.